I'm good. How are you?
With the readers, they're being communicative.
Two of them. One of them had easy ones, so I don't think she needed any questions. Haven't gone through all of that. Talk to them at all about something.
I really want to start planting.
Okay. What? Yeah. This is lecture 14. It's not Deja-vu. Sorry about that. For those of you who want to know, we will do our best to get the midterms completely graded by Friday. I'm not 100% sure if that's going to happen, but that's our goal here. The Scantron part is, of course, already done. That's easy. It's the fill-in part that takes a little bit longer to grade and to try and make it as fair as possible. So they read the answers carefully. And anyway, I'm hoping that it'll be done by Friday and we can upload grades by then too. And whenever you talk about a test, the system suddenly... all right. All right, so yes, it is lecture 14 today. And what we're going to do is continue on discussing fungal diversity as well as get into some discussion of interesting and diverse symbioses involving fungi. I think we stopped right as I was getting into the phylogeny of fungi. I'm just going to go into it again, and then we're going to go reasonably quickly through the different groups in order to get to a discussion of the biology of some of the different lineages. So again, there are 6 major lineages outlined in the book, and as you will see, At the end of today's lecture, there are other lineages that are not included here, just like with the bacteria and archaea and microbial eukaryotes. They're listing the 6 best understood, best characterized different groups within the fungi. It appears that 2 of these lineages may not be monophyletic. The details are still a little fuzzy with some of the phylogenetics that has been done, so we're going to refer to them as a group, but it may be that the Chytrids and the Zygomycota are not actually monophyletic groups. You should learn this tree. You should learn the names of the different groups. You should learn the parts that we know, how they relate to each other. So, for example, we'll talk about them in a little bit. The Ascomycota and the Basidiomycota are in this lineage called the Dicaria, and you should know how these synapomorphies map onto this tree. It's all in the book. You can read it in detail to go over that. But you should learn what's in this tree, and you should also learn what's in this table, which summarizes the scientific name, the common name if it's available for a group, and then sort of a key feature of each of those groups. And again, you should learn these by reading and studying in the book. And I'm not going to go into them in detail. So I'm just going to walk you through a little bit of an introduction to the different groups, and we're going to get through this in part without a lot of information about the groups, because we're going to talk in more detail about the biology of different representative fungi rather than focusing just on the phylogeny here. So the first group we're going to talk about are the Microsporidia. Microsporidia are all obligate, all the ones that are known are obligate intracellular parasites of animals. There are something like 1,500 species that have been described. I'm sure there are many, many more that are out there. Every time someone looks for a new Microsporidia, they frequently find them. So it's just, there's a lot of diversity there, not yet very well understood. They are incredibly small, their cells, hence the name Microsporidia. And for many years, they were classified as protists. In fact, some phylogenetic studies suggested they were among the earliest branching off eukaryotes, and that was of interest because they don't have what looks like, at least in microscopy, a traditional mitochondria. They do have another organelle that's called a mitosome, and those mitosomes appear to be derived from mitochondria, but when people originally sort of looked at them and saw that in the original analyses, they might be deeply branched within the eukaryotic tree. This was, consistent with the possibility that they had branched off before the evolution of mitochondria. That's now clearly incorrect. These are very clearly embedded within the fungal lineage. They have chitin, one of the synapomorphies of fungi, and they have many other features that they share in common with some of the other fungal lineages. And again, more detailed molecular sequence data, in part whole genome sequence data from some of these organisms, or at least many, many different genes from throughout their genomes, was needed in order to figure out that they were in fact fungi. And then many other aspects of their biology made more sense, like the presence of chitin now could be explained relatively easily because they're in a group where chitin is a synapomorphy. Again, the Chytrids are the second group. There's still a bit of a discussion about whether or not they're monophyletic, but we're going to just treat them as a group for here. And some of their features they share in common. If they're not monophyletic, the features that they share in common are most likely ancestral features of the whole clade that were then changed on this branch here so that their Chytrids are different and may have also been changed on the Microsporidia lineage. If they are monophyletic, again, it's not entirely clear, then some of the features that they have in common might actually be synapomorphies for the Chytrid lineage. Chytrids are best known as the causative agent of some nasty infectious diseases going on in a variety of environments, such as the large-scale killing of amphibians throughout the Sierras and actually, in fact, throughout the globe. These chytrids infect frogs and a diversity of other amphibians and have been responsible for some of the massive declines of amphibian populations in a variety of locations. Chytrids have a couple of really interesting features, but one of the most unusual or most distinctive features is that they are the only fungi where the spores, these are the reproductive cells, and I'll show you a life cycle in a second, they actually have flagella and they swim. And this is important because for a couple of reasons. One of them is that chytrids tend to do well in very wet environments, and by having swimming spores, they're able to disperse in water rather than relying upon air dispersal. I've listed all this stuff here. You can go through that if you want to learn more about chytrids. The book, again, has all of this described in there about chytrids. There's a lot of interesting features known about them. As I said, most of them, they like water environments. Most of them are aquatic, and in part that's related to these flagellated spores and gametes. And if you look at fungal diversity, one of the ways that, one of the easy ways to distinguish different fungal groups or to get a picture of the differences between those groups is to look at their life cycle and draw out on the life cycle where how they switch between haploid and diploid phases of their life cycle and what exactly they do in terms of production of gametes, and also how long they spend in different parts of the life cycle. So this is a life cycle of chytrids, and it's you should study, this is provided in the book, you should study these life cycles and know the major differences between the life cycles of these different organisms. And again, for chytrids, one of the key features is that these zoospores, which are produced by haploid individual multicellular organisms, they are able to swim with flagella. The gametes are also able to swim with flagella. And again, this is really important for the lifestyle of these chytrids, spending a lot of time in an aquatic environment. But you should look through these life cycles. And as we get into plant biology and fungal biology in particular, some of the major transitions that have occurred in the evolution of these groups relate directly to novel innovations in terms of how they carry out their life cycle. And then the second group of fungi that may not be monophyletic are the Zygomycota. They're also known as Zygospore fungi. And I'm going to post this online. I don't know. Yeah, you can just watch.
Consisting of asexual and sexual reproductive phases. Here, we examine the life cycle of the black red mold, Rhizopus stolonifer. The black speckles that Rhizopus produces on bread are capsules called sporangia that the fungus produces through asexual reproduction. The fungus consists of a network of filaments called hyphae that penetrate the bread and absorb nutrients. During asexual reproduction, spore-filled sporangia develop at the tops of stalk-like hyphae. The sporangia and hyphae are haploid. Sporangia break open and their haploid spores disseminate. Spores travel by air to other locations and may land on suitable food sources, such as a flaky poisson. A spore germinates an elongated hyphae which was designed to absorb nutrients. As it develops, the new haploid fungus can produce more sporangia, initiating additional cycles of asexual reproduction.
So I'm just going to pause here for a second. So it's going through many stages of asexual reproduction, producing these spores, and then those spores, which are haploid, grow up into those individual multicellular organisms. They can produce spores again, release them, and keep going through these cycles as of asexual reproduction. And then in some fungi, including these, they also can go through a phase where there's sexual reproduction. That's not all fungi do this, but most of them do.
In addition to asexual reproduction, the fungus may initiate sexual reproduction. Although Rhizopus does not have two distinct sexes, it does have two distinct mating types, designated plus and minus. They reproduce sexually when adjacent hyphae of the two types release pheromones. The pheromones trigger the hyphae to grow side branches. Note that Rhizopus generally lacks cross walls between its nuclei, and most nuclei share a common cytoplasm. As the side branches grow, nuclei stream in. A cross wall.
So this is another one of the key differences between some groups of fungi is whether or not, I mentioned this before, whether or not the hyphae are completely open, or whether or not they have these barriers inside the hyphae that restrict the movement of different components in the cell. In this particular group, the Zygospore fungi have none of those barriers.
Into the end of each branch, isolating several haploid nuclei at the ends. The nuclei are now isolated in sexual structures called gametangia. These gametangia fuse, and the nuclei inside pair up. The pairs of nuclei eventually fuse as well, and the entire multinucleate structure becomes a zygosporangium. The zygosporangium develops a thick, tough wall and can remain dormant for months. The diploid nuclei within the zygosporangium then undergo meiosis, and the sporangium sprouts. The sporangium contains the products of meiosis, haploid nuclei that are incorporated into spores. When the
So for this particular group of fungi, this is the only diploid part of the life cycle. This zygosporangium prior to meiosis, it doesn't spend a lot of its life cycle in the diploid phase. And as we see other life cycles, some of them spend much more time in the diploid phase.
These spores will disperse and germinate to form the next generation of haploid hyphae.
And I'll post a link to this video you can watch about the flaky croissants whenever you want. So in the book, they have this sort of outlined in detail life cycles for each of these groups that go through sort of what's in the video and highlight some of the key different aspects. And again, what you can see in these life cycles is they distinguish in sort of color bands. So this is all of the haploid phases of the life cycle. And then down here are the diploid phases of the life cycle. And as you'll see from some of the other fungi, there's lots more going on in the diploid phase in some of them. And again, you should study these life cycles. They are key features that distinguish the different fungal groups. And I just thought I'd back up for a second and talk about eukaryotic life cycles to remind you of them and show you some simple ones before we get into more and more complex ones. So eukaryotes that undergo sexual reproduction have an alternation between haploid and diploid phases of the life cycle. The diploid individuals are 2N, that is, they have 2 copies of each chromosome. They go through some amount of growth and life, and then eventually to produce gametes. The diploid individual will go through the process of meiosis that eventually produces cells that have 1 copy of each chromosome, labeled N instead of 2N, and then those go through some phase of their life cycle. Again, this varies between the organisms, and eventually 2 gametes will fuse together by fertilization and now produce a 2N offspring again. And you go back and forth between these haploid and diploid phases. The amount of time and what the organism does in those haploid and diploid phases is how we frequently distinguish some of the different life cycles of different eukaryotes. So this, if you spend roughly equal amounts of time in diploid and haploid, and if you have a sort of multicellular phase of the diploid and a multicellular phase of the haploid, This is called generally alternation, not alteration. Alternation of generations. Alteration of generations, not alter. Got it backwards there. Alteration of generations. And then in animals, for example, the haploid phase is greatly reduced. We don't spend extensive amounts of time in our haploid phase, or we don't do a lot of biology, basically. There are no multicellular organisms here, generally. And if you have this type of life cycle where the multicellular organism is really all in the diploid phase, that's called a diplontic life cycle. And the opposite of that, if you have a haploid phase is where the multicellular organism is, and the diploid phase is highly reduced, that's called a haplontic lifestyle, for spending time in the haploid phase, for haplontic, spending time in the diploid phase, for diplontic, and spending time in both is alteration of generations. And again, if we look at this Zygospore fungi, I'm just going to go back to that figure, they spend a lot of their time in the haploid phase as opposed to the diploid phase. And that diploid phase, as was mentioned in the video just really briefly, can lay dormant for a very, very long period of time, sort of just sit there. It's, in essence, waiting for the right conditions before something triggers it to undergo meiosis and then form the haploid spores, which become the precursors to the haploid phase of the life cycle. Within this Zygomycota group, the one they showed is this very well-known black bread mold. There are many other well-characterized members of this group, some parasites. Many of them are these saprobes that I've mentioned before. It's probably, I think I'll just move on. The fourth group of fungi, their scientific name is the Glomeromycota. They're a type of mycorrhizal fungi called arbuscular mycorrhizal fungi. I'll show you this in a second. They basically spend a large amount of their life cycle inside of plants. They have a symbiosis, mutualistic symbiosis with plants in this lineage, and there are very few species that have been characterized in this group, only about 200 species total. But if you look across plant diversity, about 80 to 90% of all plants have associations with at least one of these species. So these 200 species have mutualistic symbioses with almost all of the plants on the planet. And the way the mutualistic symbiosis works basically is the plants, via their photosynthesis, they make sugars and they provide sugars for these mycorrhizae. That's the energy source for the fungus. And the mycorrhizae serve as a way to aid in the surface area and uptake of water and minerals by the plants. So they're in essence trading sugar for water and minerals. And you can't really see this in this figure, but the hyphae penetrate into the cell wall. So plant cells are surrounded by a cell wall. The mycorrhizae, these particular mycorrhizae penetrate into the cell wall, but not into the plasma membrane. So around a plant cell, there's a plasma membrane and then a cell wall, so they get in between the cell wall and the plasma membrane. And so far, I think I mentioned this before, but so far there are no known examples of sexual reproduction in this group of fungi. As far as has ever been observed, they only reproduce asexually. People have been looking for quite some time for any signature of sexual reproduction, but as far as I know, it has not been found in any of these groups. There's actually some useful fossil information about the association between these mycorrhizae and plants that may come up in the plant lectures, but you can actually get out fossil samples from roots of plants at different time points and see the mycorrhizae that are associated with those plant roots. Do you have a question or no? All right, so that's sort of the tour of these 4 other lineages. We're going to spend a little bit more time on these 2 because many of the fungi that we're more familiar with or that we use in a variety of ways in our lives are in this last major group of fungi, which is generally called the Dicaria. And in the Dicaria, there are 2 major subgroups, the Ascomycota, which are also known as Sac Fungi, and the Basidiomycota, which are also known as club fungi. And this origin of this dicarion stage is a synapomorphy for the group. So it starts to get a little more complicated here when we look inside the life cycles of the Dicaria fungi. I'm going to come back to this in a minute because this is actually specifically the life cycle of the Sac fungi, but I'm just showing you this to highlight this dikaryotic portion. So I'm just going to go back here. There's this dikaryotic portion, and what's unique about this is these distinction where you have this process called plasmogamy, which is actually fusion of the cytoplasm between cells, but where the nuclei do not fuse together. So you have 2 cells coming together. The plasma membranes fuse, but then you have multiple nuclei inside that fused cell, distinct nuclei. And it looks something like this if you zoom in on it. If this was one of the lineages was, let's just say, mating type. This is for sexual reproduction. So let's just say there was a red mating type and a blue mating type that's shown up here. When they undergo plasmogamy, the plasma membranes fuse, but the nuclei still remain distinct. And those distinct nuclei are known, the cells with these distinct nuclei are known as dicaria. And that's a unique feature of these 2 lineages. It's a synapomorphy for this whole dicarion group. Sometimes these are also called heterokaryon, and maybe written up N plus N to distinguish it from that life cycle I showed you before with haploid shown as N, diploid shown as 2N, and so now it's not the same N. So diploid, it's 2 copies of each chromosome. This is now just to show that it's distinct, is sometimes written as this N plus N. And then after that occurs, you have a few aspects of the life cycle that happen in this dikaryotic stage until eventually the nuclei fuse as well. And that's known as karyo, kary for eukaryote for nucleus, gamy for fusion. So karyogamy occurs later. In other fungi, the nuclei and the plasma membranes basically fuse at the same time. So this whole lineage of fungi has added an extra stage to its life cycle by separating out the stages when plasma membranes fuse versus when the nuclei fuse. And there's lots of interesting things that these organisms have been able to do based upon having this dikaryon life cycle. And it depends on which lineage that you look at within this group, what they do with it. So for example, in the sac fungi, the dikaryon phase of the life cycle is pretty short. In the club fungi, it can last months, years, even decades in some of them. This part, as you'll see when I show you the life cycles of actually the individual groups, there's a little bit of difference between how the groups deal with this. And again, you should study these life cycles in the book. So one of the 2 lineages of these dicaria are known as the Ascomycota, also known as sac fungi. There's incredible diversity within this group. It includes saltwater, freshwater, terrestrial species. A large number of species have been described. There are probably tens to hundreds of thousands more that have not been described. Half of all of those that have been described are lichens. We'll see more about lichens in a few minutes. And the name comes from these sacs that they produce, which are also called asci, which contain the sexually produced haploid spores. And I'll show you this on the life cycle in a second. And the hyphae in all the dikarya are these septate hyphae, as opposed to the hyphae that don't have these divisions in them. So I think I may have posted with the slides, I'll post the whole series of this animation here, which was provided from the people in the book, but you can walk your way through the life cycle where each of the haploid lineages produce a mating structure. So here are two different mating types, plus and minus. They produce these mating structures. Those mating structures come together, and you have this plasmogamy process where, again, each individual, you get one nucleus, one cell basically from each of the two mating types coming together, and the plasma membranes fusing, but the nuclei not yet fusing. And so if you look inside here, you see always a blue and a red nuclei in one partition, and then another blue and red nuclei in another partition, and so on, with this plasmogamy process. And this is the dikaryotic stage of the life cycle. And they can grow in this dikaryotic stage of the life cycle to produce a variety of structures. In this case, the sac fungi produce these things called fruiting structures, also known as ascoma. And then you get these individual asci. These are the sacs that come out of this structure. And inside those, this is still the dikaryotic stage, you have one nucleus from each of the two mating types in these sacs that are attached to this fruiting structure. And then eventually the nuclei then fuse. And that's the karyogamy, and this is technically the stage of the life cycle known as fertilization. So they've basically separated out, again, the plasma membrane fusion from the nuclear membrane fusion and added a stage of the life cycle. The nuclei get fused here, and then those nuclei finally undergo meiosis and produce haploid spores. Those haploid spores can then asexually reproduce by mitosis inside this ascus still. So now you have asexual reproduction of the haploid spores inside this ascus, and then eventually those are released. They're called ascospores, and those can then grow in whatever environment. If they have the right conditions, they grow by, again, asexual reproduction, that is mitosis, until eventually something triggers the formation of these mating structures, and you go through it all again. So this is somewhat unusual because, again, in this lineage of fungi, they've basically invented a new stage of the life cycle not seen in many other eukaryotes. And again, you should learn these life cycles. They're an important part of the biology of these groups of fungi. So in terms of diversity, there's lots of interesting organisms within the sac fungi group. So some of them are yeasts. If you remember, I talked about the diversity of form within the fungi. There are a lot of different single-celled fungi. They're all called yeasts. It's not a monophyletic group. They're found throughout the different lineages of fungi, but one of the key yeasts to our lives is in this group, Saccharomyces cerevisiae, also known as baker's and brewer's yeasts. And one of the key things that it does is it's really good at metabolizing sugar, in particular glucose, and converting it into ethanol and carbon dioxide by this process of fermentation. And we take advantage of its ability to do this to produce all sorts of fermentation products where we either want the ethanol in wine and beer, or we want the CO2 for raising bubbles in bread and other objects. Many of these organisms in this group, not just Saccharomyces cerevisiae, are found naturally out in the environment. So they're found all over the skins of grapes. They're found in the guts of various insects. They're also known as budding yeast because they have this somewhat strange mitosis process where they, rather than completely splitting down the middle, they actually produce a little buddy during their division process. And they have, even though they're in the Sac Fungi group, they are in the dikaryon lineage. They don't have the dikaryon stage of their life cycle anymore. They have lost that. Other members of the Sac Fungi group include these cup fungi, which includes many ones that people like to eat, like morels and truffles are within this group, and these do have the dikaryon stage of the life cycle. Molds and mildews are also in this lineage. Many of the members of this group are filamentous, that is, that when the hyphae are growing, they don't form necessarily large mushroom-like structures, but just filaments. These ones reproduce by forming these conidia, and those conidia can have very distinct colors. That's frequently how we recognize many of them, by the colors of these conidia, but there are lots of parasites in this group, and there are many, I'll come back to these when we talk about parasites later, but there are many organisms that we use for food production, Aspergillus for soy sauce, Penicillium species for various cheeses, and also that's where penicillin, the antibiotic, comes from. And then the last group within the fungi, it's another member of the dikaryon group, are generally known as the club fungi, but their scientific name is the Basidiomycota, and you'll see why they form these fruiting structures that are called Basidiomata. And this includes the standard mushrooms that you may be used to seeing in the puffballs that I showed you before. There are many species that have been characterized. There are also a variety of pathogens within this group, and there are many representatives within this group that are mycorrhizae, a particular type of mycorrhizae. So mycorrhizae is basically fungal root, or generally we use this for fungi that are associated with the roots of plants. And there's this particular type, ectomycorrhizae, in this group. So here's some sort of standard club fungi pictures. Many of the sort of familiar mushrooms to you may be in this group, and many of the fungi that grow out of inside of tree trunks that you see all over the place are in this group. The puffballs that I showed you this video of before are in this group, and many of the members of this group release spores into the air for dispersal. So again, here's the simplified view of the life cycle here, and we can go through the life cycle. These fruiting structures are called basidium, and I'll come back to them when we wrap our way around, but that's where the Basidiomycota name comes from. And the spores, the individual cells, haploid cells that are inside these basidium, are called basidiospores. And these basidia are an important part of the dispersal of spores in this group. Again, like with the sac fungi, some members within this group have thrown away some of the different stages of the life cycle, but I'm going to show you the general pattern here. So these come in different mating types. Some of these lineages can have many more than 2 mating types, but we're just going to call them plus and minus for simplicity. So you have release of these spores. Those spores then grow. You get hyphae for each of the individual lineages of these spores. They come together, probably attracted by pheromones like in the other group. You have this plasmogamy process so that in the part where they're connected together, so here's the separated plus lineage, separated minus lineage, and then they fuse together, and you get the plasma membranes fusing, but not the nuclear membranes. And you get that dikaryotic mycelium. That's what you see these fruiting structures here on these gills get lined with these basidia. Eventually, the nuclei fuse, and that's, again, karyogamy, also known as fertilization. And you produce these diploid individuals. And those diploid individuals undergo meiosis to produce haploid individuals that eventually grow to form these Basidium, and we go through the cycle again. So again, the Basidiomycota, like the Ascomycota, have this extra phase of their life cycle where they can be dikaryotic. Some of them have lost that phase, but many of them still retain it, and it's an important part of the biology of many fungal, of many members of this group. So again, you should study those life cycles in the book. And what I want to do now, for the last 13 or so minutes, is start to talk about the biology of different fungi and some examples in particular of ways that they interact with other organisms or interact with us, with humans. So the first thing I'm going to talk about are parasitic fungi. Remember, the definition of parasitism is one member of a symbiosis benefits, the other one does not. And I don't know if you're familiar with the zombie ants that are out there. These are like the coolest things in the world. They're a little gross, but they're really cool.
80% of all insects live in jungles. Few are more successful than the ants. There can be 8 million individuals in a single area. But jungle ants don't have it all their own way. These bullet ants are showing some worrying symptoms. Spores from a parasitic fungus called cordyceps have infiltrated their bodies and their minds.
These fungi take over the behavior of the ants and manipulate the ants in a variety of ways to help disperse the fungal spores.
The infected brain directs this ant upwards. Then, utterly disorientated, it grips a stem with its mandibles. Those afflicted that are discovered by the workers are quickly taken away and dumped far away from the colony. It seems extreme, but this is the reason why. Like something out of science fiction, the fruity body of the cordyceps erupts from the ant's head.
Very dramatic.
It can take 3 weeks to grow, and when finished, the deadly spores will burst from its tip. Then any ant in the vicinity will be in serious risk of death. The fungus is so virulent it can wipe out whole colonies of ants. And it's not just ants that fall victim to this killer. There are literally thousands of different types of cordyceps fungi, and remarkably, each specializes on just one species.
Because they have to show it from every possible angle. All right, that's enough. There's a really good TED talk from this year's TED by a guy named Ed Yong that talks about parasites that take over the minds of their hosts. And this is one of the examples. I'll post a link to his TED Talk. It just came out a couple of days ago. These are examples of these other cordyceps species. Again, they manipulate the behavior of the organism they infect, usually arthropods, and then cause them to get, basically climb up to a point where they're high enough up on like a branch or a tree such that when they release the spores, they will get dispersed large distances. Amazingly, in many places, these are highly sought after as snacks or as sort of medicinal, traditional medicines. Figure if it can manipulate the behavior of some poor insect, maybe it can do something good for us. So there's lots known about them in part because there's lots of demand for them, so there are people collecting them from all over the planet. This is not that unusual. There are lots of examples of parasitic fungi. You don't have to worry about the facultative versus obligate here. It's just sort of distinct categories of parasitic fungi. But there are just literally thousands and thousands of examples of parasites and pathogens in the fungal lineage. There are some well-known that invade inside plants. Their hyphae grow inside the interior of plants, and then they can steal sugars and other nutrients from the plant from the inside. There's another one that's very well-known called Trichophyton rubrum. It's an Ascomycota. It feeds on keratin. For example, this species is the causative agent of things like jock itch and athlete's foot and the nail infections that many people get, and ringworm. So this one individual species causes lots of problems because it's able to live off of keratin as basically its sole source of nutrition. And keratin is in skin and finger and toenails. Stachybotrys is the black mold that you frequently see in buildings. It grows on cellulose, like the material in drywall, and it can cause, it's probably the causative agent of some of the things known as sick building syndrome, although it may not always be the case, but it produces severe allergic reactions in many people and has detrimental effects on health, and it basically will feed. It needs a little bit of moisture. Like I said, fungi really need to have some moisture in order to not get desiccated. But when a building gets wet, say during construction or after a flood, then this black mold can grow on various places where there's cellulose. The causative agent of the bat white nose disease is also another fungus. So there's lots and lots and lots of parasitic fungi that cause all sorts of problems. Oh, I took this last year. This was in Social Sciences 1100, just before class, with some big mold infestation on the building there, but hopefully that's gone. Aspergillus, another Ascomycota, also feeds on lots of things, but one of the things it feeds on are nuts, and if it grows on particular nuts, it can produce this toxin as a byproduct of its growth called aflatoxin, which is a causative agent of things like liver cancer. There are aspergilli that are used in positive ways too, not just pathogens, but many of the aspergilli are pathogens. And this is similar to what I mentioned with bacterial pathogens before. It's not that all members of a particular group of fungi are pathogenic or all are mutualistic symbionts. There are some examples of that, as I mentioned with these arbuscular mycorrhizae. But in many other lineages, there are species that, one species in a genus that's pathogenic and another species that has no detrimental effects and is, used in a variety of ways. This is just a close-up of the spores of Conidia. So again, there's lots of examples. Encephalidozoon is a Microsporidia. It doesn't usually infect people, but if their immune system is suppressed, such as by HIV infection, they can become infected. The Central Valley fever, which is a big problem in particular in farming areas, is caused by this Coccidioides, which is an Ascomycota. Again, there's just lots and lots of pathogens in the fungal lineage. And there are also a diversity of mutualistic associations among fungi. I've already mentioned some with these mycorrhizae, and I thought I would just, at the end here, talk about a few other ones. One of the most interesting mutualistic symbiosis involving fungi are what are called lichens. So these are examples of different kinds of lichens. Lichens are a very interesting type of organism that's basically emerging between two lineages. So a lichen is basically sort of a general term, although you'll see there's one phylogenetic group in particular that is involved in this, where there's a fungus involved in this interaction and some photosynthetic microbe. In some cases, that's a cyanobacterium. that is a bacteria that can do photosynthesis. In some cases, it's an algae, a eukaryote that can do photosynthesis. And together, this symbiosis has produced these organisms that in particular are able to survive really, really extreme conditions. You find them, for example, all over sort of rock surfaces in places like Antarctica. And the way they work is basically you have these layers where there's an upper layer and a lower layer from the fungus, and made up of fungal hyphae. And then on just below the upper layer of the fungus, there's a photosynthetic layer where the photosynthetic microorganism, whatever it is, either a bacterium or a eukaryotic photosynthetic species, form this layer, and then there's sort of a loose, connected with hyphae in the middle, but it's very loose, not densely packed. And that's basically how a lichen works. And the photosynthetic member does photosynthesis, makes sugars that feed the fungi, and the fungi provide access to water and nutrients and also protect the photosynthetic member. It's very similar to what mycorrhizae do with plants. The anatomy is very different, but in the plant symbiosis, the plant, like the photosynthetic member here, makes sugars that feed the fungus. That is, in the plant case, those are the root mycorrhizae. And the fungus here, and also in the plant association, provides increased access to water and nutrients. That's the mutualism, the mutualistic association that you see here. There's lots and lots of human cultures have used lichens for a variety of purposes. For making dyes, for example, a lot of traditional dyes have come from various lichens. Many people eat lichens. I've never done it myself, as far as I know, so I'm not sure what they taste like, but there are a lot of places that eat lichens. Again, you're walking around somewhere that's on mostly rock surfaces, there's not much growing, and you're bound to encounter lichens. It's useful to have something to eat. And then, as I was mentioning, many plants have this association with mycorrhizae, and mycorrhizae is basically a fungal root. It's an extension of the plant root systems with a symbiosis involving various species of fungi and the plant that, again, works in a lot of ways like the symbiosis that I just talked about. There are a diversity of kinds of fungi involved in these symbioses. Some of them are like the ones I talked about, where they're inside the cell wall but not inside the plasma membrane. Others are on the outside of the surface of the roots, and others are everywhere in between. And we'll talk about these more on Friday. Yeah.
Why is the fungus have why would that be chosen by natural selection? What are the advantages?
So there's a lot of papers on trying to figure this out. So when you have, say, mating type plus and mating type minus, and then their hyphae come together, if you have these partitions, you keep the nuclei separate initially, as opposed to what other organisms would do, which is the nuclei would fuse, and you would get two ends inside there. One of the theories about this is that when you have everything together in one mix, you can get conflicts between some of the products that are made. So this is like one organism can be dominant over the other organism, like with eye color or with all sorts of phenotypes for diploid individuals. And what this does is it allows a little bit more diversity of functions. So you can have some things that are exported and then mix inside the cell, and they function like a diploid. But other things, especially processes that are going on inside each individual nucleus, so like the regulation of individual genes, they don't get controlled as much by the other genes in the system.
That's only for some period of time.
Well, some of them it can be months they spend as a dikaryon stage. And that's where on the, I didn't really discuss this, but on the bottom of the mushrooms when these dikarya form, those can sit there for months. And there may be, I mean, there are lots of arguments about this. That's just one of the possible explanations for why delayed fertilization might be useful. And I'm happy to send you other ones. But,
Thank you.
there's, nobody agrees. Yeah.
Like you said, there's some condition that would trigger their, like, I guess, the next cycle, the next cycle occur. Then what are the triggers?
It's different in different groups. So one of the things is access to water. So because they're so dependent upon access to water, they frequently wait until they're sure that there's lots of water around. Others are with annual cycles, like the seasons. Some of them are dependent upon, like the ones living inside another organism. They're dependent upon the organism dying so that they can then get out of the organism. So they wait basically for it to stop moving and die, and then they grow up and out, and that's when some of the events happen. So it's incredibly diverse. Every type that you look at has a different trigger, in essence. Very similar to, when, for plants and animals, right? I mean, it's not conceptually that different.
Thank you. And is the flagella in the spore with the flagella? Is that flagella considered significant within the group?
It isn't. It's on the slide, but I didn't mention it. It's actually considered ancestral, and it's thought that all the other lineages that don't have it lost the flagellum. So if you go back and look at the one slide that I had on the epistoconts lineage, it's thought that the swimming spores, gametes, was an ancestral trait and then was lost by many of the descendants.
Thank you. And
Yeah.
sorry, one last
Yeah, no, go ahead. That's
Do
fine.
this to you later.
Yeah, okay.